Hello everyone, welcome back to my class. I hope you all are doing well. Today I am going to give you some short notes and 3 long questions from the chapter Principles of Inheritance and Variation of class 12 Biology. So the first question is, write short notes on A. Incomplete dominance. Answer. In the post-Mendelian period, it was observed that the law of dominance was not universally true. Several cases have been recorded where a dominant character does not mark completely the expression of the recessive characters in F1 hybrids. Therefore, the F1 offsprings show a blending of paternal and maternal characters. This phenomenon is known as incomplete dominance. Example of incomplete dominance. Incomplete dominance is well evidenced by the F1 hybrids produced in the cross between homozygous dominant red-flowered snapdragon plant R and homozygous recessive white-flowered snapdragon plant smaller. In this cross, the F1 hybrids appear neither with red nor with white flowers, but with pink flower R smaller. The heterozygous offsprings of F1 generation always produce pink-colored flower. The F1 hybrids then produced F2 progeny with identical phenotypic and genotypic ratio of 1:2:1. Here I include a figure which shows incomplete dominance in snapdragon flower. Number B. Codominance. In certain cases, both the dominant and recessive alleles lack their dominant and recessive relationship, and both of them have the capability to express phenotypically in the heterozygous condition. But in heterozygous condition, the dominant and recessive alleles remain side by side. The heterozygous offspring of F1 hybrids produce F2 offspring in the phenotypic and genotypic ratio of 1:2:1. Example of codominance. The phenomenon of codominance is found in cattle. In a particular breed of cattle with a genotype of W expressed phenotypically in white coat color, while the genotype of w into a red coat color. In a cross between homozygous white-coated cattle and red-coated cattle, the F1 hybrids are found to express a phenotypic character of reddish-gray or roan color. The roan-colored cattle of F1 generation donor possess hair of intermediate color between red and white hairs. The cross between F1 hybrids produce F2 progeny with phenotypic and genotypic ratio of 1:2:1. This figure shows codominance between white-coated and red-coated cattle. C. Backcross. The cross between F1 hybrids and either of the two parents is known as backcross. When F1 hybrids are crossed with dominant parents, all the F2 individuals express dominant character. On the other hand, when F1 hybrids are allowed to cross with recessive parents, the individuals of F2 generation with both the parental characters will appear in equal proportion. Example: backcross between heterozygous F1 hybrid tall T small t and homozygous tall T plant, only tall individuals are produced, but only 50% of them are homozygous tall T and the rest 50% are heterozygous tall T small t. This figure shows Mendel's backcross experiment in pea plant. And phenotypic and genotypic ratio of Mendel's backcross experiment is 1:1. D. Test cross. The cross between F1 hybrids and homozygous recessive parents is called test cross. The test cross is performed to determine whether the individuals exhibiting dominant character are homozygous or heterozygous. Example: cross between heterozygous F1 hybrids tall T small t plant and homozygous recessive dwarf parent plant small t small t. This figure shows Mendel's test cross experiment in pea plant. In test cross, 50% of the progenies are heterozygous dominant and 50% are recessive. Thus, 1:1 phenotypic ratio is maintained. The genotypic ratio is also 1:1 in monohybrid test cross. Thus, in test cross, the phenotypic and genotypic ratio are same. In dihybrid test cross, the phenotypic and genotypic ratio of test cross progeny is 1:1:1:1. Question number 2. How sex is determined in men? Answer: sex in human is determined by XX-XY type of sex determination. The human females bears 44 autosomes and 2 X chromosomes in diploid cells. They produce 100% similar gametes, each of them containing 22 autosomes and 1 X chromosome. Hence, the human female is homogametic. The human males are heterogametic because they produce two types of gametes. 50% bears 22 autosomes and 1 Y chromosome, and the rest 50% bears 22 autosomes and 1 X chromosome. When the egg is fertilized by sperm bearing Y chromosome, a male offspring will be resulted, and if by sperm bearing X chromosome, female offspring will be resulted. The two sexes will be produced in equal number in the ratio of 1:1. Here I include a figure which shows XX-XY type of sex determination in human being. Thus, it is evident that it is the genetic makeup of the sperm that determines the sex of the child. It is also evident that in each pregnancy there is always 50% probability of either a male or a female child. It is unfortunate that in our society, women are blamed for producing female children and have been ill-treated because of this false notion. Question number 3. Describe the genetic disorders of human beings caused by gene mutation. Answer: any disorder or abnormal character caused by defective genes or abnormal chromosome structure or number is called genetic disorder. Genetic disorders can be grouped into two categories: A. Mendelian disorders and B. Chromosomal disorders. Mendelian disorder. These are mainly due to alteration or mutation in a single gene. These disorders may be dominant or recessive, and these are transmitted from one generation to the next following Mendel's principles of heredity. Mendelian disorders may be A. Autosomal disorders. These are caused by defective genes located on the autosome. Examples of autosomal disorders are cystic fibrosis, sickle cell anemia, and phenylketonuria. Sickle cell anemia. It is caused by an autosomal mutant allele SBS of a normal allele SBA in homozygous condition. SBS. This results in the production of an abnormal hemoglobin. Due to presence of abnormal hemoglobin, the normal shape of RBCs is changed and they assume elongated, filamentous and sickle-celled forms. Such RBCs show a short lifespan. Since they clump together and often cause obstruction in the blood vessels. This causes severe anemia and sometimes even death of the patient due to damaged heart, kidney, spleen and brain as a result of clogged blood vessels. Biochemical investigation shows that globin protein of hemoglobin consists of 2 alpha chain and 2 beta chain of amino acids. In sickle cell anemia patient, the amino acid in 6th position is substituted by amino acid valine instead of glutamic acid. Phenylketonuria. It is characterized by the presence of concentrated amount of phenylalanine in blood due to lack of an enzyme called phenylalanine hydroxylase, which is required to convert phenylalanine to tyrosine. This abnormal metabolic disorder is caused by homozygous recessive autosomal genes. The patients of phenylketonuria have light body pigmentation and are physically and mentally retarded. B. Sex-linked disorders. These are caused by defective sex-linked genes. The genes located on X or Y chromosome, which control bodily characters, are called sex-linked genes. Examples of sex-linked disorders are hemophilia, color blindness, thalassemia, etc. Hemophilia. Hemophilia is a sex-linked disease characterized by the fact that the blood coagulation time is greatly prolonged, causing excessive bleeding even from the small injury. Thus, bleeding once started does not stop in the natural way and can lead to the death of the person. This disease was first reported by John Cotto. It is very common in the royal families of Russia and England. For example, Queen Victoria and her further generations were having the hemophilia disease. The genes for hemophilia is found to be associated with X chromosome and is recessive in nature. Thus, a single recessive gene produces the disease in male, and while two recessive genes are required to produce the disease in female. B. Chromosomal disorders. These are caused due to absence or excess or abnormal arrangement of one or more chromosomes. Some of these are: number 1. Genetic diseases due to autosomal numerical changes. For example, Down's syndrome. Down syndrome. It is also known as Mongolism or trisomy 21. The children with Down syndrome suffer from mental retardations. They have short and broad neck, short and stubby hands and feet, and a wide gap between first and second fingers in the feet, flattened nasal bridge and protruding tongue. This disorder was first described by Langdon Down in 1866. Number 2. Genetic diseases due to sex chromosomal numerical changes. Example: Klinefelter's syndrome and Turner's syndrome. Klinefelter's syndrome. Persons with Klinefelter syndrome are found to contain 44 autosomes, 2 X chromosomes and 1 Y chromosome, 44A plus XXY. Hence, they possess 47 chromosomes instead of normal 46. The persons with this syndrome have short stature with round head, furrowed tongue and partially open mouth. They have broad palm and underdeveloped genitalia and sparse body hair and idiocy. Physical and mental development is retarded. The genetic cause of Klinefelter syndrome is due to non-disjunction of XX chromosome. When an abnormal egg with XX chromosome is fertilized with a sperm containing Y chromosome, the zygote possessing XXY chromosomes give rise to this disease. Turner's syndrome. The persons with 44 autosomes and 1 X chromosome, 44A plus X, suffer from Turner's syndrome. Turner syndrome victims are phenotypically females. They possess 45 chromosomes instead of normal chromosome number 46. The patient has feminine characters but with rudimentary gonads. They have short stature, sealed chest, and prominent webbing of the neck. These females are sterile and their intelligence is also below average. Question number 4. Write about the genetics of ABO blood groups in men. Answer. Human blood is categorized into 4 types or groups: A, B, AB, and O, on the basis of presence or absence of specific protein substance on the red blood cells. These substances are of 2 types: antigen A and antigen B. A person is said to possess blood group A if his blood contains antigen A. Similarly, group B contains antigen B, and group AB contains both antigen A and B together. A person with blood group O has neither of these antigens. The blood group character is controlled by a set of 3 genes or alleles. The gene responsible for production of antigen A is denoted by IA. Antigen B is denoted by IB, and gene for absence of both the antigens by IO or small I. Gene IA and IB both dominant over IO or I, but not over each other. Out of the series of 3 alternative forms of genes, one individual carries only 2 genes, one from each parent. Genes IA and IB are fully expressed together in presence of the other. This condition is known as codominance. On the basis of presence or absence of antigen A and B, the classification of blood types are shown in the following table. Blood group phenotype: type A, antigen present A, genotype IA, IA or IA-IO. Type B, antigen present B, genotype IB, IB or IB-IO. Type AB, antigen present A and B, genotype IA, IB. Type O, antigen present nil, genotype IO, IO. If blood group of a couple are known, the possible blood group in their children can easily be detected as shown in the following table. The prediction can be frequently used as evidence to settle cases of disputed paternity. The blood group predictions are useful in medico-legal application. Here I include a table which shows possible blood groups of children for known blood group of the parents. Blood group of parents A and A, possible blood group in children A and O. Blood group of parents A and B, possible blood group in children A, B, AB and O. Blood group of parents A and AB, possible blood group in children A, B and AB. Blood group of parents A and O, possible blood group in children A and O. Blood group of parents B and B, possible blood group in children B and O. Blood group of parents B and AB, possible blood group in children A, B and AB. Blood group of parents B and O, possible blood group in children B and O. Blood group of parents AB and AB, possible blood group in children A, B and AB. Blood group of parents AB and O, possible blood group in children A and B. Blood group of parents O and O. Possible blood group in children O. That's all for today's video. Thank you.